Okay, it looks like our attendee list is kind of stabilizing. Good afternoon. My name is Dr. Eric Toring. I'm the Senior Vice President for the American College of Veterinary Preventive Medicine, and I'd like to welcome you to the webinar today. Our webinar is sponsored by the American College of Veterinary Preventive Medicine and our partner organizations, the National Association of Federal Veterinarians, the American Association of Food Safety and Public Health Veterinarians, the National Association of State Public Health Veterinarians, and the U.S. Animal Health Association. So with that, it is my pleasure today to introduce Dr. Renata Reimschuessel. Dr. Reimschuessel earned her VMD from the University of Pennsylvania in 1981. Upon graduation, she spent 5 years in small and exotic animal practice before obtaining a PhD in pathology, specializing in aquatic animal pathology from the University of Maryland. She served for 10 years as the Associate Professor and Director of the Aquatic Pathobiology Center at the University of Maryland School of Medicine, then joined the FDA's Center for Veterinary Medicine in 1999 to launch its aquaculture research program. There, she played a pivotal role in the 2007 scientific breakthrough identifying the cause of history's largest pet food recall. In 2010, Dr. Reimschuessel created and led the Veterinary Laboratory Investigation and Response Network, or VetLearn. This network of over 40 veterinary diagnostic labs provides collaborative capacity to investigate animal food, and drug contamination. In 2020, she led an effort to successfully validate veterinary test methods for SARS-CoV virus for VetLearn and the USDA National Animal Health Laboratory Network labs testing for animal and human COVID-19 cases. Renata has over 125 publications in peer-reviewed journals and has authored multiple book chapters. Since retiring from the FDA and VetLearn in 2020, Dr. Reimschuessel is an avid contra dancer, enjoys Irish music and beer, and occasionally plays the fiddle. So welcome, Dr. Reimschuessel.
Thank you so much. I just want to mention before I get into the details of my talk that I'm presenting this information, but it really is a cast of thousands who contributed to the work that I'm going to be describing. A little outline of what I'm going to be talking about, the 2007 pet food recall, how it started, understanding the mechanism of action, risk assessment, scientific responses. And then I'll touch on the 2008 infant formula event, which happened in China, and describe what happened and some of the responses there and World Health Organization and risk assessment. So what happened in 2007? First, it was the Ides of March. Menu Foods contacted FDA and said they're going to initiate a recall, and it was 60 million containers of their cuts and gravy style food. And this was over 100 brands that they were recalling. And the reason they were doing this was because there were animal deaths and renal failure, and that they were not sure why, but they had correlated it with imported wheat gluten from China. So they had a colony of cats that they would ask to do taste testing. They'd give 2 different kinds of foods to each cat, and they would weigh the food going in and weigh it out and know which one the cats preferred. And one of their cats died, and it was an elderly cat, and so they didn't think much of it. And then another cat died, and then another cat died, and they ended up having 19 cats die in very controlled conditions and knew that this was alarming. And they investigated what foods, they knew exactly what they had eaten, and were able to correlate it with this wheat gluten from China. When the recall hit the news media, consumers were extremely upset, obviously. Animals were dying out in the field as well. And In a matter of 6 weeks, FDA received over 18,000 phone calls. This is in 2007. There's no online reporting. Normally, FDA would get 5,000 to 6,000 calls a year. And so the complaint coordinators were totally overwhelmed. You can just imagine what the call wait times were. If you've ever tried to call the government for something, it was incredibly bad. And animals were dying of renal failures. So local vets were also trying to submit reports to CVM. They were necropsy data coming in from various hospitals and vet schools, and it was a major event. What was I doing at the time? I was in charge of aquaculture research at FDA and looking at fish, developing fish disease models for aquatic pathogens, drugs for aquaculture. And then also we did a fair amount of dosing of fish to provide tissues that had illegal drugs or chemicals in them for residue chemists to develop methods. You might say, why would you do that? Well, there were, and still are, a lot of drugs that are used overseas that are not approved for use in the United States. So we need to have actual methods that can detect those chemicals as well as their metabolites. So it's got to run through the animal in order for them to be able to detect and understand the depletion of these compounds and to make sure that we don't get any fish into the country with harmful residues. So I was involved in that research. Prior to coming to FDA, as you heard from Eric, I was a practicing veterinarian. I ran an aquatic diagnostic path lab at a medical school, and I also was doing research using fish as models for human diseases and teaching the medical students human pathology. So I had a varied background. And when this all happened, management asked if I could help with the pathology reports and also interact with the veterinary pathologists. This slide is going to be a complex slide, and it gives you the sequence of events, and it all happened extremely fast. Menu Foods came in on the 15th and told us about the recall. By the 19th, they had narrowed it down to a Chinese supplier. They were the US importer and pet food manufacturer, and pet food had been made with this contaminated wheat gluten. 2 weeks later, we found melamine in the food, and at that point, we weren't sure what really that meant, but that's when it was found, 2 weeks after the recall. About a month after the recall started, we had realized that there were actually 2 Chinese suppliers, 5 US importers, 14 pet food manufacturers. So a lot of pet food had been made with the contaminated material, plus scraps from pet food manufacturers. So things that weren't used or things like, bags fall on the floor and break open and it's not deemed adequate to put into the pet food chain. Those materials, which are presumably edible and good, were sent to feed ingredient manufacturers. One day later, the 12th of April, we find out also that 2.5 million chickens had already eaten products made by these feed ingredient manufacturers that had gone to the feed mills. And also some of the scraps had gone to hog farms, and 58,000 pigs had eaten this material. And then also scraps had gone to rendering plants. One day later, on the 13th, we found out that the US importer had sent material to a Canadian feed manufacturer. And the Canadian feed manufacturer made fish food, and that was sent back to the United States. And 106 hatcheries had used that food already, and 2 aquaculture farms with fish intended for human consumption had also been using that food. And this day, actually, the 13th of April, you'll hear in a few other slides. So this is where in the cycle you can remember where we were at as an agency for what we knew. And then the 18th, so, it's about a month out. The cycle continues, and we find out that, yes, indeed, the animals a lot of them had gone to slaughter. And in addition, some of their offal and bone meal had gone to rendering plants and back to feed mills. So it was a major event, very complex. And most of this material was found out by inspectors going to each of these places and looking through files, paperwork, bills of lading, and trying to trace where all this material had gotten to. Huge tasks. I mentioned in March 30th that the melamine was found, and it was about 2 weeks after we first notified of a problem. It was found in the wheat gluten. It was later also found in rice protein concentrate. Melamine. So what's that? I remembered melamine actually being something found in cereal boxes. Melamine plates, little plastic plates, were distributed to consumers in Kellogg's cornflakes. And anyway, it's used in plastics manufacture. The Russians also have used it as a fertilizer because it's got a lot of nitrogens in it. In combination with another similar chemical, cyanuric acid, it's made into a flame retardant. And because it can form these sheets and micelles, it's also been considered for drug delivery, for packaging drugs. So FDA and Cornell found melamine in the pet food and in cat urine. And so they thought, well, maybe this is what's causing the problem. But toxicologists rapidly said, oh my, no, it's not really toxic. And they said the LD50 of melamine is approximately the same LD50 as you would have for table salt. So it really, we weren't sure what was going on. And also, we weren't sure why it was in there. Why would it be in wheat gluten? So going back to what melamine is, it's a triazine, which means it's made with a six-member ring with three nitrogens. Melamine has even more nitrogens because the side groups all have nitrogens in them. Cyanuric acid has only three nitrogens, but still that's quite a lot when you think of the entire molecule. And cyanuric acid is one of the breakdown products and also a product of melamine manufacture. It goes from melamine to amylene, amylide, and cyanuric acid. And the bottom line is all these triazines have high nitrogen. And nitrogen is what you test for when you're testing for proteins. Usually they do the Kaldol reaction, which gives you an indication of how much nitrogen is in the sample that you're testing. And it's presumed that that is protein. So how was melamine causing kidney failure in the pets? That was a huge question. And one of the things that we did was April 2nd, still very early on, I attended calls with AABLD and AVMA, and the pathologists shared images of the crystals. And some of the key things that they were saying was they hadn't seen such crystals before. They're not like your typical struvite crystals or things that they were more familiar with. And also, there weren't enough crystals to cause the renal failure. So it was interesting because, as you know, I was at the medical school, and when I saw this spherulite type crystal, I said, I've seen this before. And it looked like crystal spherulites that you see in humans with gout. If you take an aspirate from one of those knuckles on the left, you'll get uric acid crystals in the fluid, and you can see them in the middle there. Those needle-like crystals can sometimes agglomerate into spherulites, and you can see those in humans with gout. So one of the hallmarks of uric acid crystals is that they dissolve in formaldehyde, which is, of course, the routine fixative. And so I was wondering a little bit about whether or not these crystals, possibly, if there's not enough of them to cause renal failure based on what they're seeing in the pathology, maybe some of them dissolve. So this was all a question. The other thing that I asked is, can uric acid cause renal failure? And yes, it can in people. There is a syndrome called tumor lysis syndrome, in which you get with chemo, you get rapid cell death of the tumor. And what happens is that means that there's a lot of purines and nucleic acids released. And the end product of those degradation of those compounds is uric acid. So the uric acid in this case precipitates in the kidney, and you get crystal spherulite formation, and you get kidney failure in these humans. The actual name of this syndrome is called uric acid nephropathy. Now, the mechanism of uric acid nephropathy is plumbing 101. It is not a cell injury. What it is precipitation of these crystals throughout the nephron. And you get enough of them, you actually plug up the little tubules, and that causes swelling of the nephron, which then subsequently compresses the vasculature. And when you compress the vasculature, you get vascular compromise. A kidney without blood is a kidney that fails. So that is what uric acid nephropathy causes. So at that point, I then did a big literature search, and I found that melamine actually can cause stones in laboratory animals. There was a national toxicology program that did a study in 1983 with rats and mice. And these animals made melamine uric acid stones. There's another study that I found that was a 2-year rat study in 95, which again, melamine uric acid stones were in the bladder. And these, when they analyzed the stones, they were 1-to-1 melamine uric acid. Significantly, also in the Agasara article, they mentioned one line in the materials and methods. They said they saw microcrystals in the kidney grossly. They don't mention anything about microcrystals on histopath. So again, whirling around in my rat's nest brain, I'm thinking, oh, maybe those were dissolved in formaldehyde, and that's why they never saw them on histosection. Importantly, however, is that the stones are the result of a combination of melamine and another chemical. In this case, it was endogenous uric acid. So, April 6th, I wrote up and submitted to management a hypothesis. I say, could the combination of melamine with uric acid, or maybe the cyanuric acid, which we know is used in manufacture of the flame retardant, could that cause crystals? And then second, is the pathology, the pathogenesis similar to uric acid nephropathy? So I submitted this 4 days after I had met with the pathologists. And frankly, it was received with, from management with a fair amount of skepticism. And that's true. I was also unsure. And so I was busy reading through any pathology reports that had gotten to the center. We got a lot of things faxed in, and I was pouring through these reports, and I found one from a vet in Florida. And she said, and this cat died, and it was in a household with multiple cat deaths. It died in her clinic, and she did a necropsy, and she said, and I submitted half of the kidney for histopathology, and I froze the other half. And I said, Eureka, somebody didn't fix the whole thing. And I called her up and asked her if she'd be willing to send me the frozen kidney. I shipped her a box of dry ice. She plunked the kidney in there and shipped it back to me. And I was thrilled to receive it. So this is a week later, April 13th. Remember that where we were as far as an agency of what we knew, what was happening. We had just found out about the fish being exposed to contaminated product. But that day I received late Friday afternoon frozen sections or frozen kidney. And what I did is something that is done a lot in fish medicine, not so much in mammalian pathology, but dermatologists will do it. They'll do skin scrapings and just look at them wet under the microscope on a slide. And that's what I did here. I sliced a thin piece of kidney off. And you can see here on the left is the medullary region, which is fairly avascular, and then the cortical region, which is a lot bloodier here on section. And put another slide on top and squeezed and then looked at it. And you can see here, there are those crystals. They're, at higher mag, they're a little fuzzy because you're looking through a lot of tissue there. But there's no doubt you can see these crystals lined up in this tubule. And upstream, you can see that there is cellular debris within the confines of the tubule. So here's my big moment. I said, okay, let's see if these guys are going to dissolve in formaldehyde. So I took a picture of a lower magnification of the slide with the crystals in there. And I put formaldehyde on top and then waited. And after about a half hour, it was getting later Friday night. And I said, well, it's just going to have to sit. And so I came back Monday morning, and you can see the crystals are gone. There's still this little bit of dirt on the slide. You can see that. But the crystals all throughout there are gone. You can still see some of the image of the tubules, but no crystals. So I felt that I had an answer. I did, obviously uric acid crystals will dissolve, boom, just about as soon as you drop, at least within a minute of when you drop formaldehyde on them. So these aren't the same, but I felt that they were susceptible to formalin dissolution. And it could explain why some of the pathologists said that there's just not enough crystals. If you think of how your local vet would be submitting samples, they'll cut the kidney in various thicknesses and put it into the formalin, and it may sit over the weekend before somebody gets it shipped off to the pathology lab, and then it'll sit a while. So different thicknesses exposed to formalin at different time periods, there could be quite a bit of variation. Obviously, they found them on sections, so deeper sections not exposed to as high a concentration of formaldehyde would still continue to have the crystals. That same day, actually that morning, we had a phone call with Procter & Gamble. And they volunteered to us that they had been investigating the effects of melamine and several of the triazine analogs in rats. And they reported data to us. They said, melamine plus the three analogs, the amylene, amylide, and cyanuric acid, at a ratio which they had found in some of their products, did not produce crystals. And so we discussed this, and I shared some of our ideas, and I shared about uric acid nephropathy mechanism. And then, scientist to scientist, naively, I'm in a different location than the folks at headquarters. I said, "Do you think you could do another experiment?" And I'm just wondering what management was what kind of faces they were making at that point. I didn't think of it at the time, but I was not thinking industry versus regulators talking. I was thinking scientist versus scientist. And they came back right away and said, "Sure, we can run another experiment. What do you want?" And I said, "Don't fix it with formaldehyde." And then I said, "Just do frozen sections." And then I also said, "Try using a 1-to-1 ratio of melamine with cyanuric acid, because in the animals, uric acid and melamine combine on a 1-in-1 ratio. And also, we know that cyanuric acid will combine with melamine in a 1-to-1 ratio." So 3 weeks later, they came back and said, "Yep, we found crystals in the ones treated that way." And they sent us the frozen sections, and we read the slides, and eventually wrote a combined paper, co-authored a paper on this particular study. So then, cyanuric acid. April 20th, finally, we found out that cyanuric acid was also in the pet food. It is a little bit more difficult to identify cyanuric acid, and the methods that they were using for melamine doesn't necessarily work for cyanuric acid. You have to go into negative ion mode on the mass spec, and leave it to say it's harder to find, but they did find it when we were asking them to look for it. The old literature mentions some renal toxicity. Again, it's one of these compounds considered non-toxic. And it is also generated in the manufacture of melamine. So then questions arose. Melamine and cyanuric acid, each separately, are non-toxic. What happens in the body when both melamine and cyanuric acid are ingested? How do the crystals form? So we went in vitro for this. In the lab, we've got melamine here in methanol, clear fluid. We've got cyanuric acid in methanol, clear fluid. When you combine the two together, you get a very white precipitate almost immediately. And I have a little video here that it's hard to see right now because the liquid is clear. But as I run the video, you'll see drops that go into this test tube. And with each drop, you'll see precipitate forming. And it's in real time. It's boom, boom, boom. You get the precipitate as soon as it drops. So we're thinking maybe that's what happens in the nephron when the melamine and cyanuric come together and are concentrated in the nephron. So this is a little visual of that. Two low toxicity compounds come together, they form hydrogen bonds, and they can form a crystal lattice structure that then in the body also forms these spherulite crystals. We took some of that frozen cat kidney, made frozen sections, and shipped the slide to the forensic center. They did Raman spectroscopy on the cat crystals, the crystals in the cat kidney, and also they did it on a slide. They took a drop of melamine and a drop of cyanuric acid, and in vitro on the slide, it formed these crystals. And you can see the spectrum that they got from Raman spectroscopy right here. And it is compatible with what they found with the crystals that came from the cat kidney. In the meantime, the news media was going quite actively in their investigation. They sent an investigative reporter to China, and he did some wonderful evaluations, and he spoke with people at the animal feed factories and where they were making the wheat gluten as well. And he said that they willingly told him, yeah, we put this stuff into animal feed. We can make it whatever color you want. And apparently they'd been doing this for years with melamine, but they had recently started using scrap melamine, and that scrap had cyanuric acid in it. So it was the residual from pure melamine manufacture. And some other headlines you can see here: hogs that ate tainted food to be euthanized, chickens had gotten into the food supply, fish meant for humans fed tainted food. FDA was very concerned. We were worried about all these critters that had been exposed to the contaminated feed, and we needed to conduct a risk assessment ASAP and also develop analytical methods to verify residue concentrations. So, risk assessment. This is one of those sort of dry topics, but it arises from a lot of questions and trying to come up with answers to those questions. What is in there? Okay, melamine was in there. How toxic is it? How much is in there? Those are all questions, and many of them can be answered with math. The melamine was in the wheat gluten. Okay. Then that was used to make animal feed, so there was a major dilution there. Then some of that scrap stuff was sent over to livestock feed, so even a larger dilution of that, and not everything went from the animal feed to the livestock feed. And then the questions with respect to the pig. How much is in there? How much did you eat of that pig to get an idea of what the risk is for humans? So one of the questions when you're trying to figure that out is how much accumulates in the muscle? Some of the pigs had eaten this, and some of them were at a feedlot still and hadn't been slaughtered for human consumption. So one of the questions is, some of them were there a week. How much is left in there? Does it depurate? Is it gone? How much is left? And so those were questions that we had trouble answering because we didn't have methods to look for those compounds in the muscles. So that was one of the weaknesses of doing a calculation-only risk assessment. However, based on how much came in and how much could have gotten into the pigs, due to all of the dilution factors, they calculated that a 132-pound person would have to eat 800 pounds of contaminated meat daily. And I don't remember exactly how long, but you'd have to eat a lot in order for any risk to be there. So that was reassuring, but of course we wanted to have more information. And help from industry and veterinary partners was essential in this case. And I should also add academia. We had help from multiple partners. Crucial for a rapid response. And also it was the driving force to help our develop our veterinary laboratory investigation and response network, or VetLearn, which was funded in 2010. And really, that's where I pivoted my career from fish into food animal medicine and pet food medicine. Okay, so we did quite a bit of follow-up work. We used fish as a model to study crystal formation and histopathology. And we used fish to provide tissues containing residues to validate the methods. Why did we use fish? like I said before, we had a protocol on hand that allowed us to give various illegal compounds, chemicals to fish in order to provide residues to our residue chemists working on methods. And so all we needed to do was amend the protocol to add the various triazines we wanted to use on them. And um, also, aquaculture food had been contaminated, so fish was a real issue for us, and we wanted to know what kind of residues and depletion occurred in fish. So we started, this is lightning speed for the government. We started dosing experiments within 2 weeks of finding out that fish feed had been contaminated. And we dosed with melamine alone, with cyanuric acid alone, and with both at 400 milligrams per kilogram of each drug. So that's a whopping big dose, but it was that big because that was what was found in some of the cat foods that had been implicated in animal deaths. We used fish. Later in the summer, we used pigs and chickens, but initially we did the studies in fish. How do you dose a fish? Well, we used these tubes where we put some, you can see in the lower left hand, we put fish food in there. And then we weighed the doses, milligram per kilogram. We weighed each fish and we dosed them on a milligram per kilogram basis with separate drugs in separate capsules. And then plugged it with a little bit more fish food, a nice dollop of KY jelly so that it would go down smooth. We put it into their gullet and flushed it in with tank water, which is what they were drinking. Pigs, they got a cocktail of chocolate pudding with the various contaminants, and they were quite happy to eat these treats. So the results of the studies. With the fish, dosing with melamine alone, no crystals. Dosing with cyanuric acid alone, no crystals. Dosing with melamine and cyanuric acid, many crystals. And you can see the histopathology there as well as the wet mounts. The pigs, same type of story. The kidney on the left is one from either melamine or cyanuric acid or control. The other kidney on the right-hand side is one of the treated with melamine and cyanuric acid. They, as you see, the whole kidney is swollen and it compresses the capsule and it looks blanched because there's minimal blood supply left after it swells that much. And you can actually grossly see these clusters of crystals here in the papillae. So there was a lot of work done, but basically this stuff was written up and I submitted it actually in September 2007, but it didn't get published until September 2008. But we were happy to get that part out, the part with the pet food recall, and we felt a sense of closure. But then, one day later, you can see the headlines were the news said melamine was in infant formula in China. And I was really taken aback. It was very high as 5,000 parts per million in the infant formula. The primary contaminant, though, was melamine, pure. Very, very, very little cyanuric acid. The babies that were drinking this stuff as their sole diet made stones, which were melamine and uric acid. So it's endogenous uric acid combining with this high amount of melamine being ingested, just like the rats that we described in the NTP and Gassara articles. And the problems were urinary obstruction or asymptomatic. Of course, any of you who've had kidney stones would, or know someone who has it, these can be extremely painful if they start moving. And some of these infants were just unconsolably crying And you can just imagine how upset and how deadly that could be because you couldn't tell you what was wrong. They were diagnosed with ultrasound. One hospital had 40 infants with stones, and that was one of the things that made them begin to look into the formulas because they said this is totally unexpected. And so what was happening? Melamine was being added to watered down milk by dairy farmers, and there were arrests with melamine in possession. And they basically said, hey, these two guys came with this powder and they said it's protein powder and it'll make your milk better. And so we used it. And also, melamine was being added to watered down milk. Sometimes already doctored milk was being then doctored at the producers. And arrests were made with melamine in possession at producers. The one thing that we're not sure about is, was melamine powder actually added to powdered infant formula products? There were some very, very high levels in powdered infant formula, but there's no direct evidence for addition. At that point, Dr. von Eschenbach was opening FDA office in Beijing in November 2008. This all came to light in September, and he put together a delegation which included me, and we spent 2 days in the air for 2 days on the ground, but we shared our data with about the fish and the other animals that we had dosed with melamine and cyanuric acid and those with melamine only. And I brought a whole binder of all printouts of all of the literature that I had collected so that they wouldn't be starting from scratch. The kids had stones, not crystals. And this is an ultrasound where you can see these things. These can be big. They also mentioned some sand-like crystals or stones, but they're not the melamine cyanurate crystals. This is a stone taken from a little girl in Spain. She was adopted from China. She had been born in 2007, and the stones analyzed melamine uric acid. You can see it's 1.2 centimeters, so it's something you can hold in your hand, versus the crystals, which were part of the pet food recall. You can see in size, they're way, way, way different. We're talking centimeters versus microns. So to compare the two events, 2007 pet food, 2008 infant formula, 2007 primarily dogs and cats, 2008 babies. In 2007, they were using scrap melamine, which had the cyanuric acid. In 2008, it was pure melamine. It was not the scraps, thank God. 2007, animals were dying of kidney failure due to intrarenal crystal formation, intranephron crystal formation, and the hallmark of that was death. The kids luckily developed kidney stones, the melamine urate stones. They could have no signs or pain. 6 of them were confirmed dead from urinary obstruction. That could be underreporting, but the 300,000 are the ones that were diagnosed by ultrasound. These stones were radiolucent, so they were hard to diagnose too. The 100 to 1,000 animals that died of renal failure in the pet food recall event, that's an estimate and it's hard to get at, but those are the numbers that have been published in the literature. The 300,000 were the ones that were positive, the kidney stones, and way more were screened. This was also a worldwide event in that milk products from China, for example, candy, was sold worldwide. Here, there's a headline with them finding it in Connecticut. In England, they had some interesting chocolate goodies that also were found to be melamine positive. Zoo animals that were on Chinese infant formula ended up with kidney stones as well. Again, they're sucking down this infant formula as their only food product. So FDA also did a risk assessment in 2008, and also World Health Organization had a meeting in Ottawa, Canada, which I attended. They asked me to provide background papers on melamine and the toxicology and provide data for that we had gotten in our studies during that 1 year. And it was really a remarkable meeting, and a lot of really good people trying their best to figure out what the risks are. A lot of subsequent studies. We did more studies in fish. We did studies in pigs, looking at no observable adverse effect levels. Rats, we worked with the National Toxicology Program in Arkansas, and it took them about a year before they could get the rat study started. NTP funded one of those. We did studies with neonatal rats and studies evaluating cell injury markers. So lots of that. A lot of lessons learned. Toxicity due to mixtures can be deadly. Physical versus cellular mechanisms of toxicity. Don't poo-poo the physical aspects. Melamine plus cyanuric acid. Every critter that we tested it in makes crystals. There are dose, matrix, and timing effects of melamine and cyanuric acid. You give melamine early, and then a day or so later, cyanuric acid, you can still get crystals. Melamine alone, we did some studies here, and never say never. There's a long story about this, and we were looking at why did Ogasara find crystals in the gross sections, and that I'll talk about some other time, or share literature with you on that. Bioaccumulation seems to be a minimal risk. It gets peed out rather readily, and that's why the kidney is the target organ. And newborn rats are less susceptible. And maybe that's because they don't quite concentrate the urine as much as the older ones do. Lessons learned at an agency level. Preparedness surveillance is essential, and preparedness for economically motivated adulteration is really important. Communication is important. IT, having electronic ways to submit reports was really important. Partnerships were essential, and regulatory authority. Consider the economic impacts when consumers no longer trust that regulations are followed, or that they no longer believe that the leaders of FDA are competent. So, long-term effects, even 8 years after the melamine infant formula event, people were dumping milk in China because they didn't trust, and they didn't trust their government. So, I'd like to open it up for questions now. These are my kids. They're in their 30s now. One of my favorite quotes, there's no finer investment for any community than putting milk into babies, by Winston Churchill.
Okay, so questions that have come in, we'll go ahead and I'll start with those. One of the first question is, is there any benefit to melamine as a feed ingredient or its use, or was it a disingenuous act to artificially elevate a protein content in order to financially gain from that?
It was economic adulteration. I mean, I've got to go carefully on this. Melamine, this incident and the infant formula were economic adulteration. They were cheating the system. They put it into the wheat gluten to fake the protein content. However, if you know, in Cattle feed, you can put urea into cattle feed because it has high nitrogen, but you have to say what you're putting in. But in this case, it was total economic adulteration.
And kind of linked to that, I mean, did the exporter um, that shipped it here to the U.S., I mean, did they have any idea that the product was toxic or the?
I don't think so. I think there's a big backstory about this too. When we first had our fish analyzed, when we were dosing fish for the residue depletion, our chemist came back and said, "Well, in your controls, you've got some melamine in those." And not as much as what we were dosing, but then we went back and analyzed archived feed that we had, and melamine had been coming into the country in products prior to this, but they were using the pure stuff. It's when they converted to the scrap, and nobody knew about that. They didn't know that that was toxic. They thought they would get away with it. It's just cheaper.
Okay. Um, since this has occurred, and you mentioned that 10 years after they were still dumping milk, do you know if there were any ramifications uh, in China, either government or commercial?
Big story on that. Yeah. Uh, they quite a number of people, I think in San Lu, went to jail, and I think there were maybe some executions there. At the same time, in 2007, with the at the time of the pet food recall, um, the their FDA commissioner was also arrested uh, for profiteering, and it wasn't necessarily related to the pet food recall, but taking bribes and throwing a blind eye on various food safety issues. And I think he was executed.
Okay. Back on the, when all of this first started, when the kidney failures were first being identified in cats, um, were there any other theories, before you figured out the melanine or melamine cyanuric acid combination?
Yeah, one of the there were several different things that had been put forward, but really it happened very quickly, and pretty soon we were able to document that the crystals um, uh, were the cause, and were actually there in quite abundant numbers to cause kidney failure. So, all that happened within about a month. One of the things the toxicologists really didn't want to let go of was cellular toxicity mechanisms. And with the crystals, they're spiky. They're going to damage the epithelium of the nephron. And sure, you're going to get a lot of cell markers of injury and ill cells. But in this particular mechanism, it's the actual obstruction that causes the kidney damage.
Okay. In your opinion, how common or rare in today's world are cases of intentionally adulterated commercial foods?
Well, it's interesting. When I was looking through what, the subsequent years, certainly before 2016, when I had that one slide, there were cases in China where they were repackaging some of the contaminated milk products to resell them. And there were cases in other countries where they were trying to use up this material, and they showed people with guns burying this stuff in huge holes because they didn't want people to be able to get at it and try to reuse it or use it for some other fraudulent means. You know, there there are people who will take advantage of any system. Right now, there is a big recall on infant formula right now. What is it? Um, by heart with botulism as a problem. Now, that's not intentional. However, if you go back and look at all of their safety records, maybe there is some um, uh, hiding of appropriate procedures or doing inappropriate things. So, you know,
And I mean, my, uh, thoughts are that, this was kind of um, it initiated quite a few changes in our system. It woke some folks up, if I can use those words. And I think, which speaks to one of the questions is, the oversight that we have today in feed ingredients and other products being brought in from other countries. Is the oversight much better now than it was at that point in time?
it's a good question. Way back, I mean, looking at this in, more of the human human story and political, people have been faking protein content in foods for years. In the United States, there was a manufacturer that was adding urea to wheat products. It's gone on, and I think people are more careful about that, and I think surveillance is important. But political winds shift, and if suddenly people say, "Well, we don't really want to do all this surveillance. We don't want to spend the money on it," it will come. It will come again.
Well, and you mentioned also it may come from external or it may come from internal. And that was the next part of that question is, is there, the same level of testing in domestically produced product as there
Right.
is from that we receive from, importation?
It's interesting. FDA has never really allowed Chinese infant formula in the country in the past. And the only infant formula that got in was stuff that people brought in their own suitcases entering the country. But you're right. You have to be vigilant about product that originates in the United States as well. There are people always going to be who take shortcuts and and shortcuts could be unintentionally causing problems or intentionally and know, well, this could risk something, but I want to make a quick buck. There's going to be that in any country at any time. Hopefully, I'll never see anything like this again, though. It was pretty nasty.
Well, but as
Yeah.
you said, as time goes by, memories fade, and complacency can definitely, you know, come into the picture, and pray that it doesn't happen again. But it could be something else. In the chat very early on, someone mentioned that, they have seen warning labels on plates and dinnerware that speak, that say melamine, warn about melamine.
Yeah.
Is it, in your opinion, is it serious enough that folks should get rid of those products or avoid those products?
Melamine in a dinner plate is fine. I mean, I don't put plastics in microwaves. I put it into a ceramic dish of some sort. But as far as melamine migrating from your plate into your food, no.
Okay.
And I just did want to say also that if anybody is interested in this topic and finds articles of mine that they can't access, um, email me. You're welcome to share my email with the group, and I'll be glad to send whatever articles that are not open access. And um, you know, you can look at this presentation as well.
Great presentation, a eye-opening presentation for those that maybe didn't know this story in the past. Um, we definitely appreciate your time and expertise, and we thank you very much for giving us, again, your time and expertise in today's webinar. Um,
Certainly.
I will mention to everyone that, we can't do this without great, gracious volunteers like Dr. Reimschuessel in presenting. So if you're interested in presenting a topic in our CE webinar series, please contact us. You can email me. You can go to our continuing education menu on the acbpm.org website, and you can volunteer to present because again, we can't do this without volunteers. We will not have a webinar in December. With the holidays coming up, we usually take December off, but we are ready to start back up in January. We've got a full slate for the first few months in January, but again, we need some for later on in the spring and summer. So once again, thank you to Renata. I appreciate it very much, as does the ACVPM. I thank everyone for attending. And please have a happy Thanksgiving. And if you're traveling, please travel safe. Have a great evening.